Those are two tough acts to follow. Thank you, Alicia and Angela. I'm Charlie Alpers from USGS, and I'm going to talk about a monitoring program that USGS has implemented over the last 3 years to monitor both discharge and water quality in the tributaries of Clear Lake. There's a large number of people who contributed to this. They're all co-authors of our abstract, and I have a few paper copies if anybody wants one. The people in green are USGS, the people in blue are UC Davis, and then Angela and Gordon Beaton from our Lake County Watershed Protection Agency, or Protection District. And the funding was provided by the CNRA in cooperation with the Blue Ridge Committee. Because they're non-federal funds, USGS is able to match partially through our cooperative water program. And I'll mention that the Bureau of Land Management also contributed to this study. They funded work by USGS in Scotts Creek that slightly predates this work. So as I'll see, a couple of the gauging stations and some of the water quality monitoring is funded through BLM. So here's an outline of the presentation. I'm going to briefly go over the problems that you've already heard a lot about and the project goals. Talk about our monitoring network, which you can see on the map there, but I'll go over that in some detail. Some of the discharge records for the gauging stations, the sampling dates where we took water quality samples over the last 2 to 3 years, and which laboratory analyses have we been doing on all those samples, and then some summary conclusions. So the project problems, of course, in Clear Lake, we've heard all about today so far, the harmful algal blooms in the lake, as well as the mercury fish advisories in the Clear Lake. In the past, most of the studies of nutrients have been on unfiltered water only, but it's clear to scientists who study nutrients that the filtered component is very important in terms of understanding all the interactions and what's the most available forms of phosphorus and nitrogen. And suspended sediment data also is needed from the tributaries to help modeling delivery of sediment and nutrients to the lake. So there's also been very limited data on mercury in the tributaries of the lake over the years. So in addition, much of the modeling that USGS is doing for this project, that you'll hear about in the next session, depends on load calculations, where we look at the mass over time that ends up in the lake. And so to calculate loads, you need discharge information, continuous discharge, and then you come up with an estimate of the concentrations every day at those gauging stations to come up with loads. And so we needed more gauging stations to do that. So the project goals specifically are to collect filtered and unfiltered data for phosphorus and nitrogen and mercury species over a range of flow conditions. Again, for load calculations, you really need to sample over the whole range of conditions, low flow, too high flow. And we had to establish 40 gauging stations to get the discharge data. And at gauging stations, you can instrument them to get continuous data for temperature and turbidity relatively inexpensively. So, and there's a photograph of Kelsey Creek during fairly high flow about a year and a half ago. So here's the monitoring network. The blue dots are gauging stations maintained by the California Department of Water Resources. And there's one in Middle Creek, Scotts Creek at Ockoff Road, and Kelsey Creek near Soda Bay Road. And those three are the places where UC Davis put in continuous turbidity, actually starting in 2019. So for 5 years, they maintained those through this CNRA funding work. And then USGS have the existing gauging stations that there's two in Upper Scotts Creek. One's on the south part of Scotts Creek, one's on the main side of Scotts Creek, and the other part of the basin, and then the straight down through Ockoff Road area, and then all the way up through Tule Lake. But there was no, part of this project, there was no gauging station of the water coming out of Tule Lake and into Clear Lake. And so that was one of the deficiencies that was fixed by this project. We added a gauging station below Tule Lake, and that's a tricky location because sometimes the water flows backwards. Basically, there's backflow because of high water elevation either in Clear Lake coming up Robin Slough or down from Middle Creek, which Scotts Creek's not flowing quite yet because the water's being trapped into the lake. There could be backflow in that channel right near Highway 29 where Scotts Creek comes through. So we put in a velocity meter to figure out which direction is the water going. And so we would time our samples to make sure the water's flowing, from Tule Lake toward Clear Lake before we would take a water sample for this project. And the other USGS gauging stations that were installed, there's one on Clover Creek Bypass, which is here, right before it drains into Middle Creek. And there's one on Coal Creek near Kelseyville, and there's one on Molesworth Creek at Old Highway 53, which is a small creek, but it's indicative of an urban location. We wanted to get water quality from urban type settings. So those are the 4 new gating stations. They weren't able to go in during a water year 22. We start water years on October 1st of the previous calendar year. So water years 23 and 24, when we say we had those gating stations in place, that date actually started in October 22. During water year 22, it took a while to get permits and construction done on those 4 gating stations. So we have 2 years of record from this project. One below Tule Lake, actually, Lake County is going to maintain. They found some funding to continue that one. The other 3 new gators, I think, are going to be discontinued because of lack of funding. And the 2 gating stations at Upper Scotts Creek, there's a question right now whether VLM can fund those next year as well. So we had kind of a golden age here with 10 gating stations active during this 2-year period, but we may not have it that good for a while in the future. But each one of those did have, in addition to the 3 that UCD had turbidity, all of the other 7, the USGS gating stations also had turbidity, with the exception of the one at Upper Kelsey Creek. This is a long-term gauge funded by Lake County that and USGS that does not have turbidity. The other 6 all have continuous turbidity and the relief temperature as well. Look, so here's an example of what we call a hydrograph, just water discharge over time in the blue curve. This is for that upper gauge on Kelsey Creek at Kelseyville, the one that's funded by Lake County Water Protection District. And you can see that water year 22 was relatively dry, 23 was a pretty wet year, and 24 also had a lot of storm events. The red dots are when we took water quality samples, and typically we tried to hit all 10 stations together. This was a cooperative effort with Lake County, USGS, and most of the samples went to UC Davis's Turk nutrient lab at Incline Village, Tahoe. And so, as I said, we tried to hit the peaks. We did a pretty good job on this, but we didn't use auto samplers. It was all with people out there during the daytime. So sometimes we weren't exactly on the peak because of not being able to sample at night. But generally speaking, we got pretty good coverage. This is all 10 of the stations. You won't be able to see the details here, but one was give an indication that the coverage is very good over this 3-year period. Some cases it only shows 2 years of the data because there was no gauge in place for the first water year, but we did take samples. And the N refers to the total number of samples over the 3 years. The gray areas show previous water years, but then some of these stations go back even further, where Lake County and others have taken water samples, and mostly for unfiltered nutrients, as I mentioned. All that data was used in the load calculations. And you'll hear more about load calculations from Dina Sala, who's going to be talking about that, and there's talk about Sparrow in this room the next session, as well as the poster session she's having with Sparrow tonight. And Sparrow is a spatial model of loads for nitrogen and phosphorus that USGS team has been doing. Okay, so this table shows that we had 3 schedules of different water quality parameters that we were measuring. First of all, on the left, these are the field parameters for most of those stations. We were able to use a multi-parameter sonde that measured water temperature, turbidity, pH, conductance, and dissolved oxygen. And then that FDOM, fluorescent dissolved organic matter, I talked about this morning, if you remember that part of my talk with the colorful slides and everything about how there's excitation and emission, and it measures the quality of the organic carbon. And it's a pretty useful parameter because it does, like I said, correlate with some mercury species, but it's also just another indicator of organic carbon in the water. So the Schedule A was the most comprehensive. It included all the filtered and unfiltered phosphorus species you see in blue and the nitrogen species you see in green. It had the dissolved organic carbon, the optical properties. I mentioned that whole matrix from which you can calculate specific ultraviolet absorbance or SUMA. And then mercury species were also part of Schedule A. This was done at 6 of the gauge locations. And Schedule B was pretty much the same, except minus the mercury, and a few of the sites did not get the optical properties of the dissolved organic carbon that was said, but most of them did. And then Schedule C was just sort of the way it had always been done, with just unfiltered nutrients, and some of the sites were continued to monitor in that way. Since I'm just talking about Schedule C, I'll continue. There were about 50 to 65 samples a year that Lake County monitored for what's called the Municipal Storm Drain, or MS4 program. So they're not shown on the map, but the 3, these Lake County sample sites here, 2 out of those 3 were chosen for Schedule B. So there was some filtered nutrient data at the municipal storm drains, but there's about a dozen other sites that were typically unfiltered only. for that. So you can see that the 6 gauges that were sampled for mercury are Molesworth, Coal Creek, Lower Kelsey Creek, Scotch Creek below Tui Lake, Middle Creek Horatio Road, and Clover Creek bypass. We took about 116 water samples from the full mercury analyses, another also 150 for Schedule B, a total of over 260 samples for these complete nutrient analyses over the last 3 years. Here's an example of some of the nutrient data. These are just selected for the 2 sites on Kelsey Creek, the upper one and the lower one. Again, the color coding is the blue are for phosphorus species, and the green is for nitrogen species. And I'm highlighting TN for total nitrogen. Out of all these parameters, it's the only one where we saw a significant increase statistically with the concentration of total nitrogen from upstream to downstream. And this is a comparison we're doing. You may have noticed that several of the watersheds had multiple sampling points along the flow path. We did that intentionally in the study design. Kelsey Creek, Scotts Creek, and Middle Creek, the 3 largest tributaries. We wanted multiple sample sets along the flow path so we could look for increases, particularly if there's, for example, a land use such as agriculture in between the 2 sites. We can see if that contributed nutrients to the water quality. And so this is just an analysis of concentration. We also are doing analysis of loads. And again, you'll hear more of that from Dina. And if it's not in our presentation, it'll probably be in our report that's coming out next December, but this coming December. Let's see, these are box plots if you're not familiar with them. The center half of the data are in the boxes in the 25th and 75th percentile. This middle line is the median value, and the whiskers are some standard deviations, I believe, but roughly 10 to 90% of the distributions in the bars, and then the outliers above 90% are typically in the purpose. So again, more or less comparable water quality between these 2 sites. And we're doing a similar analysis for the sites along Middle Creek and Scotts Creek, where we have multiple sampling locations. These 2 sites are different from each other, and they're not on the same watershed, but I just put them up as another contrast of water quality conditions. On the right is Molesworth Creek, which, as I mentioned, is a kind of urban site. It's relatively small in flow, but the concentrations are quite a bit higher. You see the scale here. This is a maximum of 6 milligrams per liter, where the one on the left is 2. And the ones in the previous graph were also a lower number. So Molesworth Creek has quite a bit higher concentrations of nitrate and total nitrogen in excess of 1 milligram per liter typically, which is relatively high for nitrogen species. So even though it's not a lot of flow, it's contributing to more concentration. And it also typical of the storm drains. They have they're more urban environment and these higher nitrogen concentrations typically. So in terms of overall distribution of mercury species, on the left, the phosphorus, if we look at all the data from these over 200 samples collected in the last 3 years, about half of the phosphorus is particulate in the sense that we can compare total to dissolved, half of it passes through the filter. So the entire left-hand side of that pie chart is dissolved phosphorus. And a subset of that, about 2/3 of it, is what we call soluble reactive phosphorus that Alicia talked about. It's the most bioavailable form. It's also known as orthophosphate, and it's the one that's most bioavailable. And it makes up about 2/3 of the dissolved fraction, or 35% of the overall total phosphorus. On the nitrogen side, about 30% of on the average is nitrate, when you look at total nitrogen, and that's filtered nitrate. About 70%, the solid green part of this, the dark green, is what is called TKM. K is for Keldol, which is a chemical process that gets the organic and the ammonium and ammonia. And but almost all of that is organic. We separately measure ammonia and ammonium, and it's a very small fraction in these tributaries, only 2% of the overall total nitrogen. So most of what's in that dark green area is organic nitrogen on particles, as well as dissolved. So in the lake, just in contrast, ammonia concentrations are quite high. In fact, it's higher than nitrogen, which is kind of surprising to me, but that's the nature of these eutrophic lakes. And I'll mention it just in passing that Sulphur Lake Mine has very high ammonium too, and it's actually a hydrothermal source of ammonium, which is pretty unusual. And my last data slide, this one's a little busier, but I'll walk you through it. It's the only one about mercury, which, as you know from this morning, is something I'm also concerned about. total mercury that passes through the filters, we compare now to dissolved organic carbon on the left. They both use the same size filter. It's actually a 0.3 micron filter, a little smaller than the typical filters that are used for nutrients, which is 0.45 micron. But it's essentially the same kind of thing. And there's a good correlation between filtered mercury and dissolved organic carbon. And this is thought to be because there's actually a complexation. The mercury is bound to the organic carbon chemically in that filterable fraction. And so the relationship's improved even more if we use this absorbance of 254. This is something that we measured by doing that optical properties I mentioned, the colorful matrix, the heme spectrum. We get that 254 absorbance, and it makes an even tighter relationship with carbon. So that's another reason that we like measuring that. It gives us another good way to understand the interaction between organic carbon and mercury in these systems. The two creeks that had the highest dissolved organic carbon and filtered mercury were Cold Creek and Molesworth Creek, the urban site. So I can now summarize our findings. It's pretty much just reiterating a lot of things I just said. About half the phosphorus is filterable, and about 2/3 of that is SRP or orthophosphate. Nitrate's about 30%. The rest of it's mostly organic, very low concentrations of ammonium and ammonia, in contrast to what's in the lake. The urbanized areas had high concentrations of phosphorus and mentioned species compared to the other sites, and we also had higher suspended sediment, which I didn't show any data for, but trust me on that one. And also higher turbidity that goes with the suspended sediment. But there's relatively low discharge. It'll come out of the modeling you'll hear about in the next session, but the watershed modeling and the loads modeling, just how important are those small creeks for overall loadings. I think they're probably pretty small in terms of overall sources of sediment and nutrients as well. Even if they're slightly more represented than their flow, the flows are so much smaller than Middle Creek, Scotts Creek, Kelsey Creek that they're probably pretty insignificant in terms of overall loading to the lake, except maybe locally. And then finally, that correlation of total mercury filtered with dissolved organic carbon, as well as the absorptions that we measured, was higher in those two creeks that I mentioned. And it's actually, those concentrations are higher than what we see in Clear Lake. You think Clear Lake's so contaminated, the concentration would be so much higher. But actually in Clear Lake, the partitioning for mercury is onto the particles instead of filtered. We actually get lower concentrations of filtered mercury in the lake, but much higher concentrations on the particles. So there's a lot going on in the mercury cycling, but this is just one piece of the puzzle. And again, we're grateful that the BRC and the CNRA were willing to fund this part of the work. It not just helps with the mercury, the nutrient puzzle, but it helps us with the mercury puzzle as well. And there's data available. The upper link is all the USGS data that's online is available through that map-based interface. And UC Davis also has a map-based interface where you can visualize their data, not just for the tributaries, but also for the in-lake sampling and monitoring that they've done. Okay, that's all. Thank you.
All right, do we have questions for Charlie?
I'd like to ask a question pertaining to the talking earlier about mercury.
Okay.
I didn't get a chance to. Yeah, no. You mentioned about Africa and more isotopes. I'm kind of wondering a couple of things. We test the most common, which are the most dangerous to humans and how they transmit it.
Okay, I'm not aware of any difference in the health effects of the different isotopes. First of all, none of them are radioactive, and they're all just different abundances. It's kind of like,
You know what methylated?
okay, so methyl's a different story. Any of the ones that methylate, yeah, anything that methylates is more dangerous. That's an organic form that's much more toxic. It bioaccumulates and it actually passes the blood-brain barrier. It's a lot of bad things about mercury, but and it accumulates in tissues, proteins and tissues of the fish. So there are those 7 isotopes. I can't remember off the top of my head which are the most abundant, but I think 199, 202, I think are among the top more.
They're not that distributed to the toxicity amongst them. Right? That's not really like put a secret to the I think they're not where it's methylated mercury and the other compounds that are free in the environment and transmitted. That kind of the transmission and how they affect humans in the life.
Okay, so basically, if I could answer the question, how is methyl mercury transmitted in the environment inside? How does it affect human health and ecological health? Okay, so methyl mercury can form typically in anoxic conditions, whether it's below the sediment water interface or in the anoxic water column. Those are the two main ones we think about. It can also happen in soils, flooded soils. Basically, anywhere you have a lack of oxygen, sulfate-reducing and iron-reducing bacteria are the ones that methylate, and there's a new genetic marker for that. I knew 10 years ago, but they kind of cracked the code on genetically how that works. And there's some other types of bacteria that can also methylate mercury. Those are the two that are actually, it's the research in Clear Lake by Fleming and others out of UC Davis that showed that iron-reducing bacteria can methylate mercury. That was the first study that showed that in the nature. So again, Clear Lake's on the forefront of this research all the way back then. But anyway, once it's formed of methyl mercury and it's an aqueous form, it can adsorb or somehow combine with algae, and that's the base of the food web. So once there's methyl mercury on and in the algae, anything that eats that algae will incorporate that methyl mercury, and it typically goes into the proteins. And through the process I don't fully understand, I'll just say it biomagnifies it. Every step up the food chain, it gets to a higher concentration by about 3 or 5 times at each step. There's maybe 5, 6, 7 steps in the food chain, and you go from parts per billion in the water, parts per thousand, parts per billion, parts per trillion in the water, parts per billion in the algae, parts per billion in the fish, about 1 billion times concentration factor. And once it's at high value in the fish, anything that eats that fish can start having toxic effects on its humans, other wildlife, other fish. So
Will it make the fish stupid too?
there are health effects that have been documented in San Francisco Bay. Cycle reproduction, things like that. Again, not being a biologist, I have to scour my brain for exactly what they're right. I know about bird effects, like not hatching eggs, and the grebes are one of the affected species, and blue herons, and some other, again, birds that eat larger fish are more susceptible to this.
This would be, just the last question, just because I would like to have a break of a few minutes, just to clarify, it's like pretty simple way about unfiltered water and filtered water, we talk about thinking and both.
Yeah.
I think of like a coffee filter, how do you, what really is the filter?
Are we doing it?
Unfiltered.
Okay, so for the nutrients, we use what's called a capsule filter, and water's pumped through it's called a tortuous path. It's just a big bunch of cellulose in a plastic thing. The water goes in on the outside, unfiltered, unfiltered, everything over 0.44 microns is supposed to be kept behind. It's not perfect, but that's generally how it's done. For the mercury and the carbon, we use a glass fiber filter. It's more of a flat thing, and we use a vacuum to pull it through. And it's a little different process, but we try to use the exact same filters to compare those results as I just did. So there's a slight difference in the type and size, pore size of the filters, but generally the correspondence is pretty good between those two, 0.3 versus 0.45 micron.